Hello from New York. My name is Sahil Parikh. I'm an interventional cardiologist here at Columbia Medical Center, and I'm delighted to be chairing the session sponsored by Concept Medical on Sirolimus Coated Balloons, the future of PAD. I'm delighted to have three outstanding panelists with me today. And without further ado, over the next 30 minutes, we'll have three talks and a nice long period of question and answer at the end. I'd like to introduce first Dr. Aloke Finn, my friend and colleague from Gaithersburg, Maryland. Aloke is a world-renowned expert on pathology of vascular disease and the molecular biology of restenosis, and he's going to start us off with his first lecture. Aloke, welcome. Thank you.
Hi, I'm Aloke Finn. I'm here to talk to you about Sirolimus Coated Balloons with Nanolute Technology, what the future holds for peripheral artery disease. I'd like to thank the sponsors for inviting me to give this talk. Here are my disclosures. So what makes up an effective DCB formulation? Well, it must deliver large quantities of drug within seconds, which distributes into the media and maintains therapeutic levels for more than 4 weeks to allow anti-proliferative effects. It must allow for rapid healing as compared to DES and must allow effective drug delivery to the target zone while avoiding non-target downstream effects. These are the currently approved drug-coated balloons for peripheral artery disease. And what they share in common is that they're mainly paclitaxel-based balloons that use a crystalline form of paclitaxel with varying doses. But why do we need a sirolimus DCB if we already have these paclitaxel alternatives? Well, the issue is that sirolimus is a standard for coronary artery disease treatment via DES, and in fact, paclitaxel is no longer used for coronary artery drug-eluting stents. Paclitaxel modifications, that is the crystalline form, mean the coating integrity and transfer of such products are variable, with substantial portions lost downstream into blood and tissues. And the loss of paclitaxel in the body remains a significant safety concern, which was further exacerbated by the Katsanos analysis published in JAHA a few years ago. You can see I've highlighted here some important studies done by our group and others, basically showing downstream findings in porcine skeletal muscle of paclitaxel causing either fibrinoid necrosis of small arterioles, as well as evidence of paclitaxel itself found in downstream arterioles. Others have shown slow flow when using paclitaxel coated balloons in the coronary artery, as well as impaired coronary flow reserve. Paclitaxel safety concerns certainly persist. I know we had the SFA study and a sub-analysis published last year in New England Journal. The FDA was somewhat reassured by the lack of a concerning safety signal, and so the data were comforting but limited in their scope. And of course, we know there's significant amounts of paclitaxel on drug-coated balloons, given they're fairly long. And you can see the milligram-based dosages shown for different doses here. Sirolimus offers potential benefits over paclitaxel. We all know that. It certainly has superior effects on anti-restenotic cascade, a very wide therapeutic range versus the narrow one for paclitaxel. Safety margin is probably 10,000-fold versus 100-fold for paclitaxel, and has anti-inflammatory properties as well. The issue, of course, is slow tissue absorption and short tissue retention versus an advantage for paclitaxel. Well, we can overcome these disadvantages using the Magic Touch Sirolimus Coated Balloon. It's basically a Sirolimus Coated Balloon which encapsulates submicron phospholipid particles with sirolimus, and that's how the balloon delivers sirolimus to the arterial tissue and maintains it over time. And in some ways, that really helps to enhance tissue reabsorption in this system, getting the sirolimus to where it needs to go with some sort of instant glue, and also extending its retention with these phospholipid particles. And that's really shown here in this nice experimental data from the pig porcine coronary artery, where we did Raman spectroscopy in the artery itself. And you can see here in the slide, the free sirolimus is much larger initially at 7 days versus 28 days, yet the encapsulated sirolimus stays fairly stable from 7 days to 28 days, allowing good pharmacokinetic curve data. And that's shown here in this graph, where you can see the sirolimus arterial wall tissue levels in the porcine coronary artery are sustained out to 60 days. And I think that's perfectly adequate for prevention of restenotic cascade. The coronary preclinical swine in-stent restenosis model was used just to show the efficacy and the safety of this system. Here we implant a coronary artery with bare metal stents, and then we wait 30 days, come back and treat that so-called in-stent restenosis with either a DCB, the Magic Touch DCB, or a plain balloon, and we collect the arteries 28 days later and examine tissue and organs as well as coronary arteries for histology. You can see 28-day histology on the ISR model, Magic Touch, Sirolimus Coated Balloon versus POBA. There really were no differences in terms of safety profile. Both arteries showed extremely good healing with well-formed neointima. No toxic effect or inflammatory reaction was seen. Now, this is not really a useful model for checking about neointimal inhibition given its low overall injury model, but you can see here that struts with fibrin were significantly greater with the Magic Touch system versus POBA, showing you successful drug transfer. Downstream findings, which is another important aspect. In this particular experiment, a total of 84 sections were examined. There was no incidence of myocardial infarction due to downstream emboli in either group. Microscopic scarring was observed in probably an equal number of sections, 2 from the Magic Touch and 3 sections from POBA, and there was no direct visualization of downstream emboli, showing you that there was no significant downstream emboli from this balloon. In conclusion, sirolimus is the preferred drug for intravascular interventions. Paclitaxel coated balloons are limited, I think, by the high rate of distal embolization and loss of paclitaxel in the body. Obviously, these are safety concerns. The Magic Touch Sirolimus Coated Balloon demonstrated successful drug transfer from the arterial wall out to 60 days with evidence of drug effect and no toxicity. Downstream emboli were minimal in the current coronary ISR study I presented. I believe this is a very promising new technology for the treatment of PAD. So thank you very much for your attention.
Aloke, thank you so much for a terrific talk to highlight the basic science. We now want to translate to the clinical application of this device, and Dr. Eddie Choke from Singapore is going to join us and share his results of the XTOSI study. Eddie, welcome. Thank you for being here.
Thank you to the organizers of Charing Cross and to Concept Medical for giving me the opportunity to give this presentation. These are my disclosures. XTOSI is a prospective, non-randomized, all-comers, single-arm trial investigating the efficacy and safety of Magic Touch Sirolimus Coated Balloon for peripheral arterial disease. 50 patients were recruited. Primary endpoint was primary patency at 6 months, defined by duplex peak systolic velocity ratio of 2.4 or less. Safety endpoint was defined as a composite of freedom from 30-day mortality and amputation, and also freedom from 6 months target lesion revascularization. The flowchart and patient demographics were previously presented at Charing Cross last year, and this is a quick recap. 50 patients were enrolled, of which 20 target lesions were femoral popliteal and 30 were below the knee. 40 patients reached the 6 months primary patency endpoint. Many patients were high risk with critical limb ischemia. Majority had diabetes, a significant were dialysis dependent, and a third had coronary artery disease. The vast majority had CLTI with Rutherford scores of 5 and 6 in 94%. In terms of the lesion characteristics, the lesions were mostly long, and a third had chronic total occlusions. 1/4 of lesions required retrograde access for crossing, and about 8% required bailout stenting. This is the key results slide summarizing 6 months primary patency. When considering the entire cohort, 6 months primary patency was 80%. For femoral popliteal lesions, 6 months primary patency was 88%. XTOSI's 88% 6 months primary patency was similar to studies that examined paclitaxel coated balloons for the peripheral vasculature, in which 6 months primary patency for paclitaxel coated balloons was 87% in the Ranger SFA trial and 90% in the Levant 2 trial. These are, of course, different trials, and comparisons are meant as a guide only. For the paclitaxel trials, it is noteworthy that the patients were mostly claudicants and the lesions were shorter. For below-the-knee lesions, 6 months primary patency was 74%. Singapacli is an Asian RCT which will be published soon. It has similar inclusion and exclusion criteria, and it examined paclitaxel-coated balloons for CLTI patients. The 6 months primary patency for XTOSI of 74% compared favorably to the 6 months primary patency of 40% for paclitaxel-coated balloons. In terms of safety, freedom from major adverse events was also favorable. This was 86% for the whole group 89% for femoral popliteal, and 84% for below the knee. In terms of clinical outcomes, there was a sustained and statistically significant improved toe pressures at 6 months for both femoral popliteal and below the knee groups to above 60 millimeters mercury. 60 millimeters mercury is conventionally the toe pressure threshold in which we would aim for because pressures above this level are associated with increased wound healing. At 6 months, there was an improvement in Rutherford scores. The majority of patients had Rutherford scores 5 at baseline, and at 6 months, the majority had Rutherford scores 0 for femoral popliteal cohort. Similarly, for below the knee cohort, the majority had Rutherford score 5 at baseline, and at 6 months, the majority had Rutherford scores 0. This is an example of an XTOSI participant with gangrene of right first toe with ascending cellulitis. Duplex scan showed significant diffuse anterior tibial artery disease, which was angioplastied, followed by application of Sirolimus Coated Balloon. The magic touch, or extreme touch as it was known at that time, was applied to the whole length of the anterior tibial artery. The duplex to the left is the index duplex showing diseased ATA with monophasic signal to dorsalis pedis. The duplex to the right is the follow-up at 12 months, which showed that the ATA had remained patent with triphasic signal to dorsalis pedis. The wound took about 5 months to heal and demonstrated the importance of keeping the inline vessel patent for at least 6 months, because frequently this is how long it takes for CLTI wounds to heal. At 1 year, the follow-up duplex in this patient showed that the ATA had remained patent with triphasic signal to the dorsalis pedis. The 1-year results of XTOSI trial are now available. At 1 year, primary patency for femoral popliteal was 78% and for below the knee was 59%. At 1 year, freedom from target lesion revascularization for femoral popliteal was 94% and for below the knee was 86%. Finally, at 1 year, amputation-free survival for femoral popliteal was 90% and for below the knee was 76%. We next examined the death and amputation rates at 1 year. There were 7 deaths in total, or about 14%, which was typical for this high-risk CLTI cohort. The majority, as expected, died from cardiovascular causes, including ischemic heart disease and stroke, and 1 died from pneumonia. In 1 patient, we could not ascertain the cause of death. There were 3 major limb amputations at 1 year. There are ongoing randomized controlled trials of Magic Touch Sirolimus Coated Balloon versus conventional standard balloon angioplasty, the future SFA and future BTK trials. Future SFA will recruit 279 patients with Rutherford scores 3 to 6, whereas future BTK will recruit 219 patients with Rutherford scores 4 to 6. Both trials are double-blind, 2-to-1 enrollment across 18 sites in 4 countries. Both will be CRO-controlled with independent core lab adjudication of duplex and QVA. Enrollment started in Q3 2020. In terms of the flowchart, eligible PAD patients will undergo standard balloon angioplasty, and if they fulfill the criteria, they will be randomized in 2-to-1 fashion to receive either the Sirolimus Coated Balloon or placebo uncoated balloon. The primary endpoint will be 6 months primary patency determined by duplex PSVR of less than or equals to 2.4. Follow-up will be for a total of 24 months. There are other ongoing clinical trials for Sirolimus Coated Balloons, including the Sirona trial, which is a head-to-head comparison of SCB versus PCB for SFA, and the Dual BTK RCT, which is also a head-to-head comparison, but for below-the-knee disease. The SirPAD study will be presented by Professor Nils Kucher also in the same session. In conclusion, Magic Touch PTA in XTOSI trial showed promising 6 and 12 months efficacy and safety in a high-risk CLI cohort with hostile lesions. These are early data in a small population and must be interpreted with caution, and we await results of ongoing RCTs.
Dr. Choke, thank you for sharing your very impressive data with us. Next, I'd like to turn to Dr. Stefano Barco from Switzerland. Dr. Barco is going to share with us his presentation on the SirPAD trial. looking at all comers for peripheral arterial disease treatment with the Sirolimus Coated Balloon. Stefano, thanks for being here.
Hello everybody, my name is Nils Kucher from the University Hospital of Zurich, and together with Stefano Barco we will present the trial design of the SirPAD trial. Just for introducing the two main new things about SirPAD is that it is a large trial with an all-comer design, meaning that we actually will enroll and randomize all patients that receive an angioplasty in any artery below the inguinal ligament, and we will testing sirolimus eluting balloon versus plain balloons. The second important thing is that we look for the first time for important limb events, meaning major amputation and urgent revascularization. We believe that this endpoint is cannot be doubted and has not the disadvantages of, for example, TVR or TLR, which is very much biased by open label studies, as we have to do these kind of trials open label. And herewith I would like to refer to Stefano, who will show you the trial design. Thanks.
Thank you very much, Nils, for the very kind introduction. Dear colleagues, it's my pleasure to provide you with an update on the status of SirPAD phase 3 randomized controlled trial, also on behalf of the steering committee members. In the SirPAD trial, we aimed at minimizing the number of the eligibility criteria, therefore aiming at a truly all-comer study. In fact, we have two main eligibility criteria. The first is endovascular angioplasty needed for infrainguinal peripheral artery disease. The second is the no participation in other interventional studies at the time of enrollment. How did we define target lesion? So the target lesion is the main lesion considered responsible for patient signs and symptoms. And in this perspective, we have also angiographic criteria to define the target lesion, namely a lumen compromise of at least 50% in any arterial segment below the infrainguinal ligament. The study groups of the SirPAD trials are composed of intervention group, Magic Touch PTA Sirolimus Coated Balloon, comparator is any CE certified and coated balloon catheter approved for peripheral artery disease, and patients will be randomized in a 1-to-1 randomization ratio. The primary efficacy outcome is a composite of two major adverse limb events, unplanned major amputation of the target limb and endovascular or surgical target lesion revascularization, only for critical limb ischemia, and this should occur within 1 year of enrollment. And of course, we have on top of that key secondary and safety outcomes, which will be then evaluated at 6 months, 12 months, or 24 months. This is an overview of the study flow. It will be a first clinical screening, so patients should fulfill the echographic criteria for enrollment, and the patient will be screened at different settings: outpatient, satellite hospitals, emergency room, for instance. And then there will be a second phase of screening after collection of the informed consent. This will take place in cath lab, and patients should then be evaluated for the presence, the severity, and the length of the target lesion. If the aforementioned criteria for the target lesion are confirmed, patient will be randomized on the same day during the same cath lab session, and then will be treated with either sirolimus coated endovascular treatment or with non-coated endovascular treatment. The primary efficacy outcome will be, as said, evaluated within 1 year of enrollment, and there are 3 predefined visits that have been planned at discharge within 6 months and within 12 months. Mortality will be also evaluated within 2 years of enrollment. The study was designed as a hierarchical study, primarily evaluation the non-inferiority of sirolimus coated balloon for the primary efficacy outcome within 1 year. But if this criterion is fulfilled, then we'll be allowed to perform 2 additional predefined analyses, which are in fact superiority analysis for the composite of unplanned major or minor index limb amputation and any target lesion revascularization. If also this one is confirmed, then we will be allowed to perform the second superiority analysis for the primary efficacy outcome within 1 year. And according to the type of analysis that will be performed, we will also evaluate different treating study population intention to treat as treated protocol. We assume a 10% event rate in both groups, and we set the absolute risk difference for the non-inferiority margin at 5%. The final sample size to demonstrate non-inferiority was set at 1,200 patients, and so far we have included 167 patients in the first 5 months. The predefined interim analysis is planned after the inclusion of the first 600 patients plus 12 months of follow-up. And with these pictures taken on the day of the first patient enrollment on 3rd of November, I would like to thank you very much for the attention, and thank you very much also to our team for helping us with the study and promoting this fantastic cooperation. Thank you very much.
Thanks very much, Stefano. And we have the privilege of having 10 minutes now for panel discussion. We've had several questions come in through the chat, and I wanted to start, Aloke, first with you. There's been several questions raised about the comparison of coronary versus peripheral arterial efficacy using Lymas analogues. Do you want to comment about that, and also perhaps some of our failed enterprises with Lymas drug-eluting stents in the above-knee segment like Scirocco and Strides?
Yeah, great question. I think that the barrier for safety is even higher in the coronary than it is in the peripheral, okay? Now, let's remember the coronary is part of a system which really cannot tolerate any downstream embolic effects, and any significant stenosis both would cause tissue ischemia and infarction. So if we see safety signals in the coronary, I really think that is likely to predict good safety for the periphery, where we have a little bit more leeway. They're bigger arteries, their downstream emboli tend to not be quite as important because the microvasculature of the skeletal muscle is probably a little bit bigger than it is for the coronary. So I would tell you that safety in the coronary can help predict safety in the larger SFA indication. And the second question was failure of Sirocco and other sirolimus-eluting stents. Some of that may have to do with the technology that was used at the time. I thought it was sort of felt in those situations that the spacing of the struts in those situations was very large. Interdistances were larger. Perhaps sirolimus didn't diffuse as well and circumferentially in those situations. So perhaps with more recent technologies, we will find success with sirolimus. And I'm really predicting it will probably eventually find its way into the periphery.
Great. And with respect to Nanolute specifically, you essentially demonstrated that the artery itself becomes its own sustained release device with the nanoparticles that are resident, and that the tissue residence of drug is about 60 days, which seems appropriate. Did you see any drug downstream in any of these studies that you did?
So I showed you briefly in a summary slide at the end the downstream particle matter. There were first very few evidence of any infarction, even microinfarction. And there were some downstream emboli still, but they were in a very, very, very small number of sections, like 2 to 3. And there were no difference between POBA and the Magic Touch system. So I thought that was very promising in terms of safety signal.
And compared to taxanes, you've done a lot of work on the taxane balloons with downstream emboli. How would you compare that?
I mean, I think this is a superior product in terms of its downstream emboli, especially to the higher dose taxanes like the Medtronic product. So I think overall, things are looking good for perhaps the launch of an IDE trial with this very soon in the United States for ISR and perhaps SFA eventually.
Well, so let's get to the trials. We've got 2 trialists with us who've done terrific work so far. Eddie, congratulations on XTOSI and executing in a very complex patient subset with excellent results. At this point, how do you incorporate sirolimus coated balloons in your practice? and what's your perspective for both the claudicant and the CLI patients that you've studied? I know we have trials coming forward, but just tell us your experience in the everyday practice use.
In terms of my, thank you, Sahil, for the question. In terms of my everyday practice, I'm a firm believer of leaving nothing behind. So I try to avoid putting in stents where possible in the peripheral circulation, which means then I will vessel prep the arteries as best as I can. And my practice is 95% CLTI patients, which means I treat multi-level disease, both in the SFA and also in complex BTK lesions. And in both these peripheral beds, I would vessel prep them as carefully as I can and as best as I can. And then I would apply Sirolimus Coated Balloons in both SFA and also BTK arteries. And after that, I will review if there is any doubt as to any flow limiting dissections. Then if I feel I would need a scaffold, then I would put in a bare metal stent. And essentially, you've got the advantages of a scaffold and also with a drug-eluting technology together to inhibit the new intimal hyperplasia. I have to say my rate of putting in stents is about 10%.
Yeah, that's phenomenal. A question came in through the chat asking about why not use paclitaxel and sirolimus, sort of double dip and maximally inhibit neointimal hyperplasia. If you have a comment on that, that's great. But I also would wonder why the comparator in some of the trials isn't a taxane balloon, because I know you have them available in your neck of the woods. So if you want to comment on either of those questions, that'd be great.
If the question means putting in sirolimus and then putting in paclitaxel, I have never ever done that. And I wouldn't, I feel, if you look at the properties of sirolimus and paclitaxel, and I think Aloke has already summarized it very elegantly. Sirolimus has clearly superior critical parameters, inhibits the proliferation of smooth muscle cells much better. It is an immunosuppressant, so it's got anti-inflammatory properties. So for me, that is enough. I guess the question, if the question means, why don't you use paclitaxel in certain times and use sirolimus in other times, then this is kind of my practice as well. And you would selectively use paclitaxel in certain places. And for me, the use of paclitaxel at the moment is limited to critical limb ischemia and in the SFA and popliteal arteries.
Definitely, I want to turn to you as we're drawing towards the end of the time period. SirPAD is a very ambitious trial with a lot of patients enrolled. When do you anticipate full enrollment? You're at a pace of about 167 or so patients at 5 months. When do you anticipate full enrollment of the study?
It must be said, we enrolled these 170 patients despite COVID-19. So in fact, we've seen many patients with acute limb ischemia, much more than anticipated. And consistently, we have also seen many outcomes over the past weeks. So in February, we are also very happy with this enrollment rate, 1.5 per day approximately. So we expect to see the final results, to have the final results in 4 years from now, and to perform the first interim analysis after the inclusion of the first 600 patients plus 12 months follow-up in 2 years.
Fantastic.
But we're also starting extension to a second or third center in Switzerland just to increase and speed the enrollment rate up.
And do you, in your practice, use primarily a strategy like Eddie, leave nothing behind?
Yeah, exactly. That was also to answer the question that was raised. So why not using paclitaxel as a comparator? But simply because it cannot be a comparator in standard of care. So we couldn't actually have it as a comparator, and we should then rely on non-coated balloon, simply because at that time, and we are talking about 1 year ago, there was some sub-signals that paclitaxel may increase 2-year mortality rate. Sure. That has been not confirmed in the Swedish trial that's been published late in 2020. But yes, our practice was also very much influenced by the type of patients we have seen, and we didn't know what to do with specific subgroups of patients, and we didn't know whether Sirolimus Coated Balloons are better or at least non-inferior as comparator, and this is exactly the reason why we decided to perform the study.
Great. Well, I'm afraid we're out of time. 30 minutes goes quickly. I want to thank each of my panelists, Aloke Finn, Eddie Choke, and Stefano Barco, for your participation and excellent presentations today. I believe we're going to be heading back to the UK to Dr. Greenhalgh, and we want to thank you for being part of this session here at Charing Cross 2021. Thanks so much, everybody. Have a great day.